Beethoven. For 20 years, scientists studied locks of Beethoven's hair to find out what killed him. In 2023, DNA proved the hair did not belong to Beethoven. It belonged to a woman no one can identify. Everything we thought we knew was wrong. And the truth, when it finally came from the right hair, was worse. Ludwig van Beethoven died on March 26, 1827, in a rented apartment in Vienna's Schwarzspaner house. He was 56 years old. The final months had been a cascade of organ failure that his physicians could document, but not explain, let alone reverse. It had begun the previous December. Returning to Vienna by open carriage in freezing weather, he developed fever, cough, and splitting pains in his side. He began vomiting. He began spitting blood. His abdomen swelled with fluid, liters of it, pressing against his organs from within. His doctors performed 4 abdominal punctures over the following weeks, draining enormous quantities each time. The first procedure resulted in infection. The wound never fully closed. A brownish fluid continued to leak from his body for weeks. By March, jaundice had set in. His feet were swollen. His urine had stopped. Hepatic encephalopathy, the brain fog produced by a liver that can no longer filter toxins from the blood, settled over him like a slowly falling curtain. On the afternoon of March 26th, during a thunderstorm, Beethoven reportedly raised his fist toward the ceiling and collapsed. He did not wake again. An autopsy was performed the next day by Dr. Johann Wagner. The liver had shrunk to half its normal size. Its surface was leathery, covered in nodules the size of beans, the unmistakable topography of advanced cirrhosis. The spleen was swollen to twice its normal volume. The pancreas was shrunken and fibrous. The kidneys were encased in a thick membrane leaking brownish fluid, their calyces studded with calcified deposits. It was a body in ruins. But an autopsy in 1827 could describe damage. It could not explain cause: alcohol, infection, genetic predisposition, toxic exposure, or some combination of all 4. That question would require tools that would not exist for nearly 200 years. And in the meantime, something else was happening. Something that would derail the investigation before it even began. People were taking souvenirs. Collecting relics from the famous dead was not unusual in the 19th century. In the days surrounding Beethoven's death, friends, admirers, and medical attendants cut locks of hair from the composer's head. Some took them as mementos. Some sealed them in lockets with inscriptions and passed them down through generations. Among those who took a lock was a 15-year-old musician named Ferdinand Hiller, who reportedly cut the hair from Beethoven's corpse the day after the autopsy. The lock passed through the Hiller family, was mounted in a locket with an inscription, and over the following century became the most famous physical relic of Beethoven in existence. It was this lock, the Hiller lock, that scientists turned to when forensic technology finally caught up with Beethoven's request. In the year 2000, a research team analyzed strands from the Hiller lock and reported extraordinarily high levels of lead. The finding was dramatic. Lead poisoning offered a unifying explanation for Beethoven's constellation of symptoms: the gastrointestinal distress, the irritability, the possible contribution to hearing loss. A 2007 follow-up study reinforced the conclusion. Papers were published, documentaries were filmed, books were written. The lead poisoning theory became the dominant narrative in Beethoven medical biography. For over 2 decades, the world believed it had answered the composer's question. It had not, because the hair was not his. In 2014, a master's student named Tristan Begg at the University of Tübingen conceived a project that would grow into the most comprehensive genomic analysis of a historical figure ever conducted. Working with William Meredith of the Beethoven Center at San Jose State University and an international team spanning Cambridge, the Max Planck Institute, KU Leuven, and the University of Bonn, Begg set out to sequence Beethoven's entire genome from authenticated hair samples. 8 locks were acquired from collections across Europe and the United States. Each was tested for DNA, assessed for damage patterns consistent with its claimed age, and compared against the others. 5 of the 8 matched. They came from the same European male, with ancestry consistent with Beethoven's documented genealogy, genetically most similar to people living in present-day North Rhine-Westphalia. The best preserved among them, the stump flock, yielded a genome sequenced to 24-fold coverage, a level of detail that rivals modern clinical sequencing. But the most famous lock, the one that had anchored 2 decades of lead poisoning research, the one on which an entire diagnostic narrative had been built, came from a woman. The Hiller lock was not Beethoven's hair. It had never been Beethoven's hair. DNA analysis identified it as originating from a female with genetic markers highly prevalent among Ashkenazi Jewish populations. Meredith speculated that the authentic Beethoven hair may have been replaced at some point with strands from Sophie Lyon, Ferdinand Hiller's wife. How or when the substitution occurred remains unknown. What is known is this: every study conducted on the Hiller lock, every measurement of lead, every inference about opiates, every conclusion about toxic exposure, applied to a woman whose identity we do not know, and said nothing whatsoever about Ludwig van Beethoven. The forensic investigation had spent 20 years studying the wrong person. With the authenticated genome in hand, the real investigation could finally begin. And what it found was not a single answer. It was a convergence, a medical picture built from layers of genetic, infectious, and toxic evidence that describe a body under siege from multiple directions simultaneously. The first finding was genetic. Beethoven's DNA carried variants in the PNPLA3 and HFE genes associated with significantly elevated risk for liver disease. These are not rare mutations, but in the right combination, they predispose a carrier to cirrhosis with a potency that modern hepatology takes seriously. Beethoven's genome did not guarantee liver failure, but it loaded the chamber. The second finding was infectious. Researchers identified hepatitis B virus in the sequencing data from the stump flock. Beethoven had been infected before his death, at minimum several months prior, possibly much earlier. Hepatitis B is a known driver of chronic liver inflammation, fibrosis, and ultimately cirrhosis. In a patient already genetically predisposed and already consuming significant quantities of alcohol, the virus would have accelerated a process already underway. The third finding came in May 2024. A separate team, led by Harvard pathologist Nader Rifai, tested 2 authenticated locks for heavy metals. The Berman lock contained 258 micrograms of lead per gram of hair. The Halmthayer lock, which Beethoven had personally hand-delivered to the pianist Anton Halm in April 1826, telling him, "Das sind meine Haare," contained 380 micrograms. Normal is less than 4, 95 times the expected concentration. The hair also contained 13 times the normal arsenic and 4 times the normal mercury. The irony is precise and painful. The lead poisoning that had been proposed in 2000 and debunked in 2023 was real after all. It had simply been proven with the wrong evidence. The conclusion was correct. The sample was not. Science had arrived at the right answer through the wrong door, been told the door was fake, and then found the answer again through the right one. But the lead was not enough to have been the primary killer. It was a co-conspirator, not the sole perpetrator. The sources were almost certainly mundane: wine sweetened with lead acetate, stored in lead-lined vessels, and medical treatments of the era that routinely employed lead-based compounds. The diagnosis is a cascade: genetic predisposition to liver disease, compounded by chronic alcohol consumption, compounded by hepatitis B infection, compounded by decades of heavy metal accumulation. Each factor alone might have been survivable. Together, they were not. But the genome held one more secret, and it had nothing to do with disease. Beethoven's Y chromosome was compared against DNA from 5 living men in Belgium who carry the surname Van Beethoven. Genealogical records traced all 5 men and Ludwig to a common patrilineal ancestor, Aert van Beethoven, born in 1535 in Kampenhout, Belgium. The 5 living Van Beethovens shared essentially the same Y chromosome, confirming their descent through an unbroken male line from Ireland. Ludwig van Beethoven's Y chromosome was entirely different. Somewhere in 7 generations between 1572 and 1770, the biological paternal line had been broken. At least one child in that lineage was not fathered by the man whose name he carried. The man the world knows as Ludwig van Beethoven was not, in the strictest genetic sense, a Van Beethoven at all. The 5 men in Belgium were informed of the finding on the evening of March 15, 2023. They were shocked. Every day they introduce themselves and see the recognition that comes with the surname. The DNA told them the connection they had always assumed was, biologically, not there. There is one question the genome could not answer, the one Beethoven himself cared about most. His hearing loss began in his mid-20s. By his early 30s, it was severe enough that he wrote the Heiligenstadt Testament, the unsent letter to his brothers, in which he described the humiliation and isolation of his condition. He wrote that he had considered ending his life, and that only his art had held him back. By his mid-40s, the deafness was total. He continued to compose the Ninth Symphony, the late string quartets, the Missa Solemnis, all of it written in silence, heard only inside the architecture of his mind. The 2023 study found no clear genetic basis for Beethoven's deafness, no variants associated with otosclerosis, no evidence of Paget's disease. The 2024 LEAD study raised the possibility that chronic exposure contributed to auditory nerve damage, but the mechanism remains speculative. Beethoven asked for one thing: that the world understand his disease. 200 years later, science has explained his liver, his infections, his poisoning, his ancestry. It has not explained his silence. The genome answered questions Beethoven never asked, and could not answer the one he did. In 1802, in the village of Heiligenstadt, Beethoven asked that after his death, his doctor describe his malady and make it public, so that the world might be reconciled to him. That request took 221 years to fulfill. The answer arrived not from a doctor's pen, but from a genome extracted from a lock of hair, kept in a letter, kept in an archive that survived 2 world wars, carrying DNA that survived 2 centuries of handling. Long enough for a technology Beethoven could not have imagined to read what his body had been trying to say all along. The forensic tools that identified malaria in Tutankhamun's blood, that matched the Romanov bones to Prince Philip's DNA, that traced the arrow in Ötzi's shoulder, those same tools, applied to 5 locks of a composer's hair, did not solve a murder. They solved a life. The Ninth Symphony was composed by a man who could not hear it, whose liver was disintegrating, whose blood was poisoned, and whose genome carried a family secret no one alive could remember. Beethoven's testament was written in ink. His genome wrote a longer one,